Hello everyone, my name is Glauco and I'm a PhD student from Londrina State University in Brazil, and I present the work Polymorphisms and Haplotype Structures from Transforming Growth Factor Beta-1 Gene show subtype-specific associations with susceptibility and clinical presentation in woman breast cancer. So this work was developed in the Laboratory of Studies and Applications of DNA Polymorphisms here at the Londrina State University under the supervision of Professors Maria Angélica Erraro Watanabe and Roberta Lozzi Guimarães. So, breast cancer is a major global health concern, being the most frequently diagnosed cancer among women, both here in Brazil and worldwide. Importantly, both its incidence and the number of deaths related to this disease are still increasing in recent years, despite the advances made in its screening, prevention, and treatment. But when you talk about breast cancer, it's important to emphasize, to keep in mind, that we are not talking about a single disease, and we are actually talking about a group of neoplastic diseases that occur in the breast. The seminal work by Sorley and co-workers used micro-array technology to identify gene expression patterns and characterized at least 4 different subgroups of breast cancer with different prognoses, namely Luminal A breast cancers, which has the best prognosis among all breast cancer subtypes, and Luminal B, HER2 enriched, and basal-like or triple-negative breast cancers, which are more aggressive forms of breast cancers with higher rates of metastasization and shorter disease-free survival and overall survival. In clinical routing, Luminal A breast cancers are classified as those expressing hormonal receptors, namely estrogen and progesterone receptors, but not overexpressing the human epidermal growth receptor 2, or HER2. Luminal B tumors are those that express hormonal receptors and show high proliferation indexes, but do not and can express or not HER2. HER2 enriched subtype is characterized as not expressing hormonal receptors and overexpressing HER2. And triple negative breast cancers are those that do not express neither hormonal receptors, thus lacking therapeutic target and showing the worst prognosis among all breast cancer subtypes. So there are several factors in tumor microenvironment contributing to breast carcinogenesis, such as immune cells and growth factors. And among these factors, TGF-beta is of crucial importance. TGF-beta is transforming growth factor beta, TGF-beta is a pleiotropic cytokine that exerts cell and context-dependent roles, and this becomes evident in breast cancer, where in less aggressive forms of cancer, such as Luminal A tumors and initial phases of carcinogenesis, TGF-beta is capable to induce cell cycle arrest and apoptosis, while in more aggressive forms of breast cancer, such as HER2-positive breast cancers and triple-negative breast cancers, this cytokine can induce immunosuppression, angiogenesis, and to promote epithelial to mesenchymal transition, or EMT, thus favoring tumor cell invasiveness and migration. The pleiotropic effects of transforming growth factor beta can be explained on the basis of its signaling pathways. So when TGF-beta binds to its serine kinase receptors, intracellular mediators MADs are activated and translocate to the nucleus, where they interact with a plethora of transcription factors to induce or inhibit the gene expression. So depending on the transcription factors that are active at the cell at the moment these MADs are activated, different responses will be generated. And also TGF-beta can activate alternative pathways such as PI3K and HasMap kinase pathways, which are associated with proliferation and survival of cancer cells. Interestingly, in tumors already presenting HasMap kinase hyperactivation, such as those overexpressing HER2, the TGF-beta pathway is biased toward this route and inhibits the SMAD-dependent route, leading to a phenomenon called hyper-EMT and increasing the invasiveness and motility of cancer cells. TGFB1 gene locates at the long arm of chromosome 19, and there are several polymorphisms with functional implications that are described for this gene. So the first of them is G-800A that occurs in a putative binding site for Krebs family of transcription factors at the promoter region of TGFβ1. We've got C-590T, which also occurs at the promoter region which also occurs on the promoter region of TGFβ1 in a binding site for yin-yang-1 transcription factor. And T allele for this polymorphism has been implicated in higher levels of TGFβ1 plasma, in plasma of patients. And this polymorphism has been shown controversial role on breast cancer susceptibility. And T allele carriers had also shorter disease-free survival in a Chinese population of breast cancer. C29T polymorphism, which occurs at codon 10, leading to a proline to leucine change in the signal peptide sequence of the cytokine. For this polymorphism, the C allele was implicated in higher TGFβ1 secretion. This polymorphism has been also associated with breast cancer in a Caucasoid population, and the C allele was also correlated with disease-free survival in Chinese population. Lastly, the G74C polymorphism, which occurs at codon 25, leading to an arginine to proline change, has been shown controversial role on breast cancer susceptibility and has unclear functions on TGFβ1 secretion. So the motivations for our study was that all these polymorphisms have been shown controversial results on the literature associating with breast cancer. These polymorphisms have been never studied in conjunction in a Brazilian population before, and previous studies associating these polymorphisms with breast cancer have never taken into account the specific effects of TGFβ1 in different breast cancer subgroups, making it a possible source of controversial results found on the literature. And further, the association with prognosis parameters in different breast cancer subtypes is completely unknown, leaving an important gap in basic cancer research field. So to form the study, we collected biological material from 323 breast cancer patients from the region of Londrina and from 405 control women. These women had no signs of mammary alteration proven by a recent mammogram test, no familial history of breast cancer, and no personal history of any other cancer during her life. So DNA was extracted, quantified, and submitted to PCR, and then PCR products were submitted to enzymatic restriction for genotype of the different polymorphisms, including the study. And fragments were all analyzed by electrophoresis in a polyacrylamide gel. Haplotype reconstruction or haplotype inference was made using the phase software, and statistical analysis consisted of age-adjusted logistic regressions for association studies and Kendall-Stahl-B rank correlation tests for correlations between TGFβ1 variants and clinical pathological features. So passing to our results, this table shows the clinical pathological features for different breast cancer subtypes. So here we've got luminal HER2 negative breast cancers, which are mainly luminal A breast cancers, luminal B HER2 positive breast cancers, HER2 enriched breast cancers, and triple negative breast cancers. As shown, the age did not differ between our control group and any breast cancer subgroup analyzed. And as expected, luminal HER2 negative breast cancers had the most favorable prognosis parameters when compared to other subtypes, as can be seen by tumor size, histopathological grade here, cellular proliferation index here, p53 mutation frequency, lymph node metastasis, and tumor stages. This table shows the genotype frequencies for each polymorphism included in the study. Here, BC refers to the entire group of breast cancer, and HER2 positive refers to samples that are positive for HER2, irrespective of hormonal receptor status. So it corresponds to the sum of luminal B HER2 positive tumors and HER2 enriched subtype, which were grouped together due to their similarity in prognosis parameters and to their shared expression of HER2 overexpression. So passing to our association analysis, we found that promoter region CT polymorphism was a risk factor for general breast cancer in dominant model. And when we stratified this analysis by subgroup, we saw that this association was specific for HER2 positive samples and not for other subgroups. So CT polymorphism, which is associated with higher TGFβ1 secretion, was a risk factor for HER2 positive breast cancers. Also, codon 10 polymorphism T allele, which is a low expression, low TGF-beta secretion allele, was a protective factor against this same group of tumors in recessive model. And codon 25 polymorphism was a risk factor for this same group of tumors and not other tumors. But this result might be viewed with caution due to the large confidence interval observed here, which resulted from the rarity of this polymorphism and the small sample size we've got to HER2 positive samples. So regarding haplotype structures, just to explain a little for those who are not familiarized with this nomenclature, haplotypes are blocks of SNPs, individual polymorphisms that are inherited together in the same chromosome. So here we've got 8 possible TGFβ1 alleles that are written in sequence from G, A promoter region polymorphism, passing through CT promoter region polymorphism, codon 10 polymorphism, and codon 25 polymorphism. So in sequence from the left to right. So we observed that HER2 positive samples showed the greatest difference from control group in haplotype frequencies, although this was not statistically significant. Then we passed to association analysis for individual haplotype structures, and we noted that GTCG haplotype was a risk factor for breast cancer, while GCCG haplotype was a protective factor against general breast cancer. And when we passed to the subtype stratified analysis, we observed that the association for GCCG haplotype was specific for luminal HER2 negative tumors, while the association for GTCG haplotype, which is a high producer allele due to the T allele from promoter region CT and to the C allele from codon 10, were a risk factor for HER2 breast cancer. Also, we observed that the GCTG haplotype, which is a low producer allele, was a protective factor for HER2 breast cancers, both in recessive and in dominant model. And then we performed correlation analysis to investigate if these TGFβ1 genetic variants could influence the the breast cancer clinical presentation. And what we found was that variants associated with high production of TGFβ1, here shown in red boxes, just like T allele from promoter region CT and GTCG haplotype, were indicative of decreased proliferation rates in luminal HER2 negative tumors, so indicating a favorable prognosis, but were positively correlated with worse prognosis parameters in more aggressive forms of breast cancer, just HER2 positive and triple negative breast cancers. Namely, they indicated higher histopathological grades and higher rates of lymph node metastasis in HER2 positive samples, and higher tumor size and higher rates of lymph node metastasis in triple negative breast cancers. The opposite pattern was observed for variants associated with low TGFβ1 production. So here we've got the codon 10 polymorphisms, which were negatively correlated with lymph node metastasis in HER2 positive samples and with tumor size and lymph node metastasis in triple negative breast cancers. And the GCTG haplotype, which carries the T allele from this polymorphism, was also shown to be negatively associated with prognosis parameters in HER2 and triple negative breast cancers. Also, GCCG haplotype, which were previously shown to be a protective factors against luminal HER2 negative breast cancers, correlated positively with proliferation indexes in the same group of tumors and with histopathological grade in HER2 positive samples. And finally, the G-800A promoter region polymorphism and its associated haplotype structures were correlated with worse prognosis parameters both in HER2 and in triple negative breast cancers. So namely tumor stage and histopathological grade. So lymph node metastasis is a severe event that occurs at later stages of carcinogenesis. And then we sought to investigate if the associations of TGF-beta variants with lymph node metastasis were independent of other prognosis parameters. So first we identified the tumor size to be the only clinical pathological feature associated with lymph node metastasis positively with lymph node metastasis in all breast cancer subgroups, is strongly associated. And then we perform a logistic regression analysis for TGFβ1 variants, adjusting for tumor size. In this analysis, we found that the promoter region CT polymorphism was a risk factor for lymph node metastasis in HER2 positive breast cancers, independently of the tumor size. So discussing a little bit the results, for individual SNPs, we found concordant results in previous literature for all polymorphisms, except for G74C, the codon 25 polymorphism. But it's important to emphasize that your study was the first to investigate this polymorphism in specific subtypes of breast cancer. For haplotypes, we found that the results globally reflected those found for individual SNPs that were represented in these haplotype structures. So we've got the low production GCTG haplotype being a protective factor against HER2 positive breast cancer. The high producer haplotype GTCG should be an indicating increased susceptibility to HER2 breast cancer, and GCCG haplotype should be a protective factor against Luminal A HER2 breast cancer. Previous literature failed to observe any association between these polymorphisms, these haplotypes, and breast cancer, but again, our study was the first to investigate these haplotypes in specific subtypes. Highlighting the importance of TGF-beta variants on breast cancer prognostication, a previous research identified that high producer alleles indicated reduced disease-free survival in Chinese population of breast cancer. And our results indicated that high producer alleles, namely promoter region CT and GTCG haplotype, should be associated with lymph node metastases in highly aggressive forms of breast cancer, such as HER2 and triple negative. So these results might be explained by the effects of TGF-beta in promoting epithelial to mesenchymal transition. During this process, TGF-beta increases the expression of CCR7 on cancer cells, and these cells then migrate through a gradient of CCL21, which is highly expressed in lymph nodes. Also, recent research have shown that the TGF-beta produced by regulatory T cells in lymph nodes increases the metastatic potential of these cells, seeding metastases to bone marrow, liver, lung, and brain, which are classical tumor metastatic sites for breast cancer. So as an overall conclusion, we found that TGF-beta variants have subtype-specific roles in breast cancer pathogenesis, with high producer alleles indicating worse prognosis and increased susceptibility to more aggressive tumors such as HER2 and triple negative tumors, while being protective against clinical pathological features in poorly aggressive ones, and the opposite pattern being shown for low producer variants. So these are consistent with the dual roles of TGF-beta on breast cancer pathogenesis and suggest these variants as interesting candidate markers for this neoplasia. I would like to acknowledge all the institutions involved and the financial support by funding agencies. Thank you.
Thank you, Glauco. Very interesting presentation. The first question we have for you is from Connor. And it is, have these genetic variants been studied in the context of other cancers?
Yes, actually in our lab, we studied these variants in other types of cancers. So we studied in breast cancer, in childhood acute lymphoblastic leukemia, and in other pediatric tumors such as Wilms tumor and neuroblastoma. And there's a vast literature investigating these polymorphisms in a varied types of other types of cancer. They are well studied. Just commenting a little bit more, the results are the same for epithelial cancers, and for LLI, it's the acute lymphoblastic leukemia is the opposite. So TGF-beta seems to indicate better prognosis in acute lymphoblastic leukemia.
Great, thank you, Glauco. Just for the interest of time, I invite you to answer the other questions remaining in the chat just directly in the chat box, okay?
Okay, thank you.
Great, thank you.